Institute, and Nicola's going to tell us about one of the resources that's been funded as part of the Human Functional Genomics Initiative, and that's the screening lab based over in Cambridge. So over to you, Nicola. Thank you.
Great, thanks, John. Can everybody hear me okay? Have I got this? Can you hear me through the mic all right?
Has it gone green?
It is green, yeah. Maybe if I move this up a bit.
There you go. Now it's green.
Okay, right. Okay, that sounds better. I can hear myself now. Okay, so I've got 10 minutes to tell you about the Functional Genomics Screening Laboratory, which exists at the Milner Therapeutics Institute in Cambridge. The whole team, more or less, is here today, so anything that I miss, the team has a poster, and Uli and Wong, the two laboratory leads, are here, and they would be happy to answer any questions you might have. So, what is the Functional Genomics Screening Laboratory? It is a collaboration between University of Cambridge, AstraZeneca, and the Medical Research Council. And it's been set up as part of the Human Functional Genomics Initiative. And the reason why it exists is to give access to UK-wide academics and SMEs and industry to arrayed CRISPR screening platforms. So you might be asking yourselves, why is it important to have developed a lab like this that can aid the functional genomics community within the UK? The main reason for that is what we actually mean by an arrayed CRISPR screen. So for those of you that are familiar with CRISPR and all the technologies that have come from it, you probably first came across it as a pooled CRISPR screen, which we do in big flasks with massive numbers of cells that we can grow at scale, and we can knock out every protein coding gene within the genome in one huge experiment. And then we can perturb that population of cells, and we can ask, how does it respond to a specific drug or a particular change in environment? And then we use next generation sequencing at the end of that screen to determine which genes, when knocked out, have helped those cells to survive or become resistant, and which genes have made those cells die faster. So it's really, really excellent for asking binary questions. And now you can combine that with single-cell RNA-seq, and you can get more granular data. And it works brilliantly for cells that you can expand massively in culture. But actually, there are lots and lots of models out there where you can't do that. You can't put them in big blasts of cells and ask those types of questions. But if you can take small numbers of cells and put them in a 96-well plate, for example, and perturb those cells within each well, you don't need to apply next-generation sequencing because you know which genes you've perturbed in each well. And in addition to that, you can use multiple endpoints to look at what's happened to those cells when a specific protein coding gene has been knocked out. So, for example, you could do a whole host of imaging endpoints if you've got a really fancy machine, which we're lucky enough to have in the lab, or you could run them through flow to ask different questions, or you could take the supernatant from those cells, put them into another 96-well plate, and then run various different kinetic and other color readout assays to understand what's going on with those cells. So you've got a much deeper detail of actually looking at the biology of the cells in each of those wells. So that's what we do in the lab. If we want to run whole genome in an arrayed CRISPR screen, that's over 200 96-well plates, or it's between 60 and 70 384-well plates. So what you've probably appreciated already is that you can't really do that by eye yourself to get the readout at the end. So you need automation platforms. And this is what the MRC have helped us to put in the lab at the Milner. So here you can see pictures of our new system. On the left-hand side here, we've got our dosing platform. So there are 2 robots in that platform. They can pick up plates from an incubator, feed it into various different liquid handling machines that we have on this platform. They can dose cells, they can dose guide RNAs into those cells. We've got an electroporator on that platform to introduce Cas9 and the guide RNAs if we're not able to use lipofectamine for a particular model. And then, after a few days of having those cells and those plates in the incubator, We can use our feeder reader platform, our Yokogawa, high content imager, to actually look at what's happening to those cells at the end. And we've also got a couple of other things on the platform. So this is our IQ, this is a fax that will work in plates very, very rapidly. So we've been able to set up this automation platform, which allows us to run these screens at scale. And this is just a couple of examples of initial data from Sean, who's in the audience, and Antigone, that have been working with this initial data. This is a experiment that's looking at efferocytosis. And there are two populations of cells stained here. Now, when I started in the lab, I was doing these kinds of experiments, and I would sit there at the microscope, I'd look in my dish, and I'd have a clicker in my hand. And I'd count the cells by hand, and I'd move on to the next image and count again and keep records of all of that. Obviously, I've just told you how big some of these screens can be. So it's impossible now to do that kind of readout at scale. So what we have is, once the images have been analyzed in the Yokogawa, we're using software to then segment those different cells and what's going on in those images and to do all the counting for us. And then we're able to look through that data and crunch that data to get a list of hits out the other side. We are not set up to run experiments for ourselves. As I said, we are working with all of you and any models that you might have to bring them in and run. And they are not going to cost you to run those experiments. We are funded either through collaboration with AstraZeneca, where there's a model that's of interest to them and they'd be interested in seeing the data. They will fund that experiment for you. If it's not so much of interest to industry or AstraZeneca in this case, we also have an access fund from the MRC that we can then also apply to use to fund that screen for you. So we are looking to you to provide model and ideas to us, and then we can assess whether we can help you to run an array CRISPR screen to move your data forward and explore more about the models and diseases you might be interested in. We are also working with SMEs and other industry partners on a fee-for-service basis. We ourselves don't approve these projects that are approved by a joint steering committee, which is led by John Mill, and we review those applications twice a year. So what do we need from you if you have a project that you think might work on this platform? We need a list of genes that you want to perturb. We need to know what your model is, what state it's in. Have you tested it in 96-well format? Do you know that you can introduce Cas9 and guide RNAs into that system? And one thing I must stress is we don't work with lentivirus. So all of the pooled screens that you may have heard about and read about, they use lentivirus to introduce both components, Cas9 and the guide RNAs. Our system is not set up to handle lenti. So we're introducing Cas9 and the guide RNAs using either lipofectamine or electroporation. So we need some initial evidence from yourselves that that system works in the model that you're interested in. And then the final point, which is really, really crucial here, we need to know what your assay endpoint is. And do you have positive and negative controls for that assay that give a nice window? Because in running these screens, they tend to generate a lot of noise by the size that they tend to be. We need a really nice positive and negative endpoint assay that gives us a nice assay window that allows us to then call hits at the end of the screen. And again, we then bring those models in, we onboard them, we do a lot of tests, we work out how it's going to run through the automation, we run that screen for you, we analyze the data, and we give you that data back. So if you're interested, either come and speak to us while we're here. We really, really like to speak to people that are interested to understand their models and their assay endpoints so we can give advice ahead of time. You can then look on Connect Health Tech. You can scan the QR code. We've got a 5-6 pager where we ask you lots of information about your model, and then you submit that to us. Oops. Once you've done that, decision is made by the Joint Steering Committee. about whether or not we can bring the model in, i.e. is it biologically relevant and is it technically feasible? We give feedback on all proposals. We don't just decline. We often give you the information that you need to take it forward so that we can bring it in. And then for the models that we do bring in, we go into a full collaboration agreement with yourselves and your university contracting office. Generally, these screens take about a year plus to run, so it's not a straightforward process. As I said, the team that we have are here, but we are hiring at the moment, so we've got 2 open positions for research associates and 1 open position for a research assistant. The research assistant position closes today, so if you're interested, speak to the team or apply online. The team are all here, as I said. We'd love to hear from you, and if anybody's got any burning questions, I can answer them now. Thank you very much.
Thank you, Nicola. Has anyone got any quick sort of technical or questions for Nicola? I mean, it really is a great setup. And there's a question at the back. But I really encourage people to talk to Nicola and the team because they will really guide you through the process really, really well.